Picture this, right? You are in an intensive care unit.
Oh, man. High stress.
Exactly. The rhythmic, just almost deafening beeping of monitors is all around you. You are fighting for your life against this unknown, rapidly spreading infection.
Right, and the clock is ticking.
Your temperature is spiking to, dangerous levels, your blood pressure is plummeting, and literally every single second matters. Now, in this ultra-modern, super high-tech medical environment, what do you think is the very first thing the doctors do to figure out what's killing you?
I mean, it would make complete sense to assume they go straight for the big guns, right? We live in the genomic age. We can map out human DNA. We can engineer targeted therapies. You'd think the first step is, I don't know, digital and highly complex.
You would think they immediately reach for some multi-million-dollar DNA sequencer.
Yeah, absolutely.
But they don't.
Mhm.
Instead, to make that immediate, life or death decision on how to treat you, they reach for a cheap 140-year-old dye test that takes just a few minutes and costs little pennies.
It is wild when you really think about it.
Okay, let's unpack this, because today our deep dive is into a biological sorting hat called the Gram stain.
Yes, the classic.
It is a seemingly simple test that divides bacteria into two fundamental camps, right? Purple or pink. Gram positive or gram negative.
Which honestly sounds like a middle school science experiment. But it is so much more than a color matching game. It is the foundational secret decoder ring for modern medicine.
Right.
Without it, modern infectious disease treatment would practically grind to a halt.
We are going to uncover the completely accidental origins of this test in a 19th century morgue, explore the microscopic architecture it reveals, look at its massive real-world implications, and of course dig into the messy edge cases that prove biology just absolutely hates neat little boxes.
Biology definitely hates boxes. And to really understand how this simple color test still dictates treatment in a 21st century ICU, we have to go back to understand how it was created in the first place.
Right, because he wasn't trying to change the world.
Not at all. It wasn't designed to sort all the world's bacteria. The person who invented it was just trying to solve a very specific, highly localized problem.
So we have to travel back to 1884, to a pathological institute in a Berlin morgue.
A fun place to start.
Yeah, right. This is where we meet Hans Christian Gram. He was a Danish physician, pharmacologist, and bacteriologist. And he was working under a man named Carl Friedlander.
Right.
Now, Gram was dealing with a massive daily frustration. He was spending hours looking through a microscope at these thin slices of lung tissue from patients who had died of pneumonia.
And this was before modern imaging, obviously.
Exactly.
Oh, yeah.
Back then, they used these ordinary aniline dyes, and the fundamental problem was that everything on the slide stained the exact same color.
Right, so imagine trying to find a specific gray pebble on a beach full of gray pebbles. It's impossible. The human lung cells, the bacteria, the background tissue, everything absorbed the dye and became a uniform visual mush.
The visual mush, I love that.
Gram couldn't see the bacteria for the lung cells. He just wanted a way to make the microscopic invaders visually pop against the background human tissue so he could, actually study them.
He wasn't trying to revolutionize microbiology or create some grand taxonomic system for all life on Earth. He literally just wanted better contrast on his microscope slides.
That's the beauty of it.
So he starts tinkering with different chemicals.
Yeah.
He develops this multi-step washing process. First, he hits the tissue with a gentian or crystal violet dye.
Okay.
Then he fixes it in place with a specific iodine solution, which, fun fact, is actually still called Gram's iodine in labs today.
It really is.
And finally, the crucial step. He washes the whole thing with alcohol.
And when he looked through the lens after that alcohol wash, he realized something incredible. Some of the bacteria held on to that deep purple dye, completely resisting the alcohol wash.
Wow.
But others, the alcohol washed them clean. They were completely colorless against the background.
Which is such a happy accident.
Really?
But it wasn't just a neat visual trick for better contrast, right?
Huh.
This accident actually settled a massive live scientific dispute right there in the morgue.
Oh, yeah. See, Gram's boss, Friedlander, had previously identified a specific bug, which we now call Klebsiella pneumonia.
Okay.
Friedlander was convinced this specific bug, which ended up washing clean and staining pink, was the sole cause of pneumonia. He thought he had solved the disease entirely, like case closed.
But Gram's new purple and pink sorting hat proved him wrong on the spot.
Exactly. By using his new staining method, Graham was able to show that many of these fatal pneumonia cases actually involved a completely different organism, one that held on to the deep purple color, making it what we now call gram-positive.
And what was that bug?
That purple organism was later identified as Streptococcus pneumonia. We now know that specific purple bug is the single most common bacterial cause of community-acquired pneumonia worldwide.
That is wild. So Friedlander was looking at pink bugs, Graham finds these purple bugs, and suddenly they realize pneumonia isn't just one disease caused by one bug. It's a condition that could be caused by two totally different culprits.
Exactly.
But here's the part of the source material that just blew my mind. Graham publishes this method in a German medical journal in 1884. He gives it this incredibly dry, modest title. Something like, I think it was like, on the differential staining of schizomycetes in sections and dried preparations.
Very catchy.
Super catchy.
Uh-huh.
But in the paper itself, he's basically apologizing for it. He calls his own method defective and imperfect. He thought it was just a preliminary tool, and he never even tried to patent it.
Right. He essentially gave away what would become the most universally used lab procedure in the history of medical science.
Right.
He didn't profit from it at all. And if we connect this to the bigger picture, that humility highlights something vital. The Graham stain was invented as a real-time referee in a live debate about what was killing patients right in front of them, not as an abstract scientific theory.
Okay, so Graham invents this test. It works. It sorts the bugs.
Oh.
But he had absolutely no idea why it worked.
Nice.
He just knew purple meant one thing and colorless meant another.
Yeah, for over 7 decades, doctors and microbiologists used this test clinically without having a rigorous physical explanation for it.
7 decades.
7 decades. It wasn't until the mid-20th century, notably through the work of a microbiologist named Milton R.J. Salton in the 1950s and 60s, that we finally got answers.
Oh, okay.
Salton and others used new biochemical techniques and electron microscopes to actually look at the physical architecture behind those colors.
So let's break down that architecture, because this is where the chemistry meets the biology. When you run a Graham stain today, it's still basically that same step-by-step process Graham invented.
Right.
First, the crystal violet goes in. At this point, everything on the slide is purple. Then, the Graham's iodine is added. Now, our deep dive sources mentioned the iodine acts as a mordant.
Yeah, think of a mordant as a chemical anchor. The iodine binds to the crystal violet molecules that have already slipped inside the bacterial cells.
Okay.
When they bind together, they form this massive crystal violet iodine complex. It makes the dye molecules bulky.
So it's like the iodine makes the dye molecules put on giant winter coats so they can't fit back out the door they just came through.
That is exactly it. They become chemically too large to easily diffuse back out of the cell. Then comes the most important step, the decolorizer, usually an alcohol wash.
Right.
This is where the physical architecture of the bacteria actually dictates the result.
Right. So if the dye is wearing a winter coat, the alcohol acts on the door frame. If it's a gram-positive bacteria, the ones that stay purple, they have this incredibly thick, densely cross-linked wall made of a polymer called peptidoglycan.
A very strong wall.
Yeah, it's like 20 to 80 nanometers deep, which is huge on a microscopic scale.
Yeah.
When the alcohol hits that thick wall, it dehydrates it. It shrinks the pores. It literally shrinks the door frame, locking those bulky winter-coated dye molecules permanently inside.
The cell stays a deep, dark purple. But a gram-negative bacterium is built completely differently.
How so?
It has a very thin peptidoglycan layer, only about 7 to 8 nanometers, sometimes just a single layer thick.
Wow, that is thin.
Right. And this thin layer is sandwiched underneath a second outer lipid membrane. That outer membrane is mostly made of fats.
So for the gram-negative cells, when the alcohol hits, it doesn't just shrink the door. Because their outer wall is mostly lipids, or fats, the alcohol dissolves that outer membrane entirely.
Exactly.
It blows the roof off the house.
Right.
And that thin little inner layer underneath offers zero resistance. The winter-coated dye molecules just wash right out into the street. The cell becomes completely colorless.
Yeah, they're just gone. And then to actually see those colorless cells, you have to add a final step, a counterstain, usually a pink or red dye called safranin.
Because otherwise you wouldn't see anything.
Exactly. The purple cells are already so dark they just ignore the pink, but the newly colorless cells soak it up and turn pink. Hence, gram-positive is purple, gram-negative is pink.
It's brilliant. Although, the sources mentioned there is actually still a bit of a debate about the exact mechanism here, which is wild to me for a test from 1884.
Yeah, it's not totally settled. Historically, the explanation was pure physical trapping, just the alcohol shrinking the pores of that thick mesh like a stopper in a bottle, just as you described.
Right.
But later biochemical work pointed out that gram-positive walls are woven through with negatively charged polymers. So the modern textbook view is a blend.
Ah, bit of both.
Right. It's a mix of the physical pore shrinking and a chemical magnetism holding on to the positively charged dye complex. It's a genuine nuance that scientists still discuss.
Which just shows how complex this simple test really is. Understanding that structural difference, thick armor versus a thin inner layer hidden by a fatty outer shield, it really explains why this exact test is still the bedrock of real-world medicine today.
Absolutely. The difference between those two types of walls isn't just about what dye they hold, it's about how we have to fight them.
Because time is tissue in a hospital. If you culture bacteria in a lab to definitively identify its exact species, that takes, 24 to 48 hours?
At least, yeah.
But a gram stain takes minutes. If a patient's blood culture flags positive for an infection, the lab instantly smears it on a slide and gram stains it. We talked earlier about doctors changing an empiric antibiotic regimen.
Which is just a doctor's highly educated first guess before all the lab data is in.
Right. So if the doctor hears the lab say gram-positive Kochi in clusters, they're immediately thinking, okay, this is likely Staphylococcus. But if they hear gram-negative rods, they're pivoting entirely to something like E. coli. That single phrase changes the drugs going into the patient's IV within minutes.
And that speed is most critical in cases like bacterial meningitis, where the infection is in the fluid around the brain and spinal cord.
That's terrifying.
It is. Meningitis can kill within hours. There is no time to wait for a 48-hour culture. The gram stain is read at the bedside or rushed to the lab as a high-stakes pattern-matching exercise.
I love how the text breaks down this visual pattern matching. It's like high-stakes detective work. If the lab tech looks through the microscope and sees gram-positive Diplococci,
Meaning purple spheres sitting in pairs.
right, purple spheres in pairs, the doctor immediately suspects Streptococcus pneumonia, a common pneumonia bug that has made its way to the brain. But what if the lab tech sees pink spheres in pairs?
Gram-negative Diplococci. That points the doctor straight toward a hyperlethal meningitis bug that requires an entirely different, incredibly aggressive public health response because it's so contagious.
Man. And if it's small, pink rods.
Then they are dealing with something like Haemophilus influenza. That visual pattern, just shape and color, drives the very first critical dose of antibiotics. We use visual morphology as a proxy for species identification when the clock is ticking.
It's not just everyday hospital cases either. The sources detail the 2001 anthrax bioterrorism attacks in the U.S. The first fatal case, a photo editor named Robert Stevens in Florida, was initially worked up using a cerebrospinal fluid gram stain.
Yeah, that's right.
The doctors looked at the slide and saw large gram-positive rods, which is the classic signature of weaponized anthrax.
Think about that contrast. You have a modern 21st century national security and public health crisis, and the initial steering of that massive federal response relied on a Victorian-era bench technique.
Here's where it gets really interesting, though, because I can hear you, our listener, asking, in an era where we can literally sequence a genome in an afternoon, why are we still relying on this color matching game?
Because the structural difference revealed by the stain directly dictates how the treatment will work.
Oh, right.
Take beta-lactam antibiotics like penicillin. Their entire mechanism is to target and block the cross-linking of that thick peptidog-lichen wall. So knowing you have a gram-positive bug tells you the primary target is exposed and vulnerable.
But the gram-negatives have that fatty outer membrane acting as a shield.
Exactly. That outer membrane is a massive barrier. To get past it, antibiotics have to slip through specialized channel proteins called porins.
Porins, okay.
Yeah. This outer membrane and its porins are gatekeepers, physically blocking many antibiotics from even entering the cell. This drives intrinsic antibiotic resistance. So the 19th century test isn't just telling you a name. It is literally showing you the physical armor the bug is wearing, which tells the doctor which weapon to draw.
That makes perfect sense. The color reveals the armor. But if the gram stain is so fundamental to medicine, is it a perfect binary system? The short answer from our deep dive is absolutely not.
Not even close?
Biology is infinitely complex.
Right.
Trying to shove all of bacterial life into two little bins, purple or pink, requires ignoring some massive acts of biological deception.
The binary is incredibly useful, but nature hates a strict binary. There are known exceptions and masters of disguise baked right into the system.
So let's talk about how bacteria hide from this test. First up, you have the heavily armored bugs, like the one that causes tuberculosis. You can't gram stain them reliably at all.
No, they're too tough.
They have a wall loaded with mycolic acids, which are these long-chain fatty acids that make them super waxy. It's like they're wearing a slick raincoat. The crystal violet and alcohol system just bounces right off.
For those, you need an entirely different technique called an acid-fast stain, which uses heat and strong acid alcohol to literally melt and punch through that wax.
That sounds intense.
It is. And what confuses people is that on the evolutionary family tree, tuberculosis bugs are actually closely related to gram-positive organisms. They just refuse to behave like them under this specific dye.
Then you have the ghosts, Mycoplasma species. They don't have a cell wall at all. So asking if they are gram-positive or negative is like asking if a ghost is wearing a thick or thin winter coat. The question just doesn't apply.
Exactly.
And then you have stealth bugs, like the ones that cause syphilis or chlamydia. They're simply too thin to see with this dye, or they can only survive inside a host's own cells, hiding from the test completely.
We also can't forget the shapeshifters. There is a bug called Gardnerella vaginalis, which is a key marker used for diagnosing bacterial vaginosis. It is officially classified as gram variable.
Gram variable, meaning it changes.
Yeah, depending on its mood or growth phase, it stains unpredictably. Sometimes pink, sometimes purple.
Wow. Even classic textbook purple bugs can fool you, though.
Yeah.
If a sample sits in a lab for too long and gets old, the bacteria start releasing enzymes that basically cause them to eat their own armor. The wall degrades, so an old, dying purple bug will suddenly look pink, giving a falsely gram-negative result.
And beyond the biology, there's the human element. The decolorization step with the alcohol is highly sensitive.
Wait, I have to jump in here. Are you saying a tired lab tech with a heavy hand who leaves the alcohol on for 3 seconds too long could literally erase the purple, causing a doctor to prescribe the completely wrong drug?
That is entirely possible.
Oh, wow.
If you overwash it, you can strip the purple dye right out of a genuinely thick-walled organism. It's a classic teaching example of human user error masquerading as a biological result.
So what does this all mean? If a bug is wearing a raincoat, or if it's a ghost, or if a lab tech just sneezes while holding the alcohol bottle, how can doctors trust this for a meningitis diagnosis where hours matter?
Well, what's fascinating here is this vulnerability is exactly why the gram stain isn't just about mixing chemicals. It requires highly trained technicians and rigorous quality control.
This is an art and a science.
Right. The medical field understands that the gram stain is a heuristic. It's a mental shortcut based on anatomy. You trust it because you train the people interpreting it to recognize the context, the shape of the bacteria, the patient's symptoms, and the potential for error.
You treat it as a vital tool, not an infallible crystal ball.
Exactly.
Before we wrap up, we should clear up a few major misconceptions our sources highlight. One of the biggest is that gram-positive and gram-negative represent two distinct branches on the evolutionary tree.
Yeah, for a long time, microbiologists assumed the purple and pink results perfectly tracked evolutionary history. But modern DNA sequencing proved that wrong.
This is where we need a good analogy. Just because a bat and a bird both have wings doesn't mean they are closely related, right?
Right.
It's convergent evolution. They
Yeah.
both figured out how to fly independently. The same goes for bacteria. Just because two bugs stain purple doesn't mean they're cousins. It just means they independently evolved thick armor.
That's a perfect way to put it. There is an ancient bacterial lineage that stains gram-positive, but it actually has an outer lipid membrane that is much more typical of gram-negatives. The color result and the evolutionary family tree don't always match up perfectly.
Another misconception. Gram-negative bacteria do not completely lack peptidoglycan. They do have that wall material. It's just very thin, so they can't trap the dye when the roof gets blown off.
Right.
And maybe the most important misconception for the general public, neither group is inherently more dangerous than the other.
That is so important. They differ in structure, not inherent deadliness. It's true that the lipid outer membrane of gram-negative bacteria contains an endotoxin that can trigger severe septic shock in patients.
Which is very bad.
Right. Very bad. But plenty of gram-positive organisms produce highly lethal toxins that are just as deadly through completely different mechanisms. They are equally capable of being lethal.
So as we wrap up this deep dive, I want to bring it back to you, the listener. The next time you are sitting in a doctor's office with a stubborn infection, or heaven forbid, You are in an ICU receiving an empiric antibiotic regimen. Remember that the life-saving decision wasn't just made by a modern computer.
It was likely guided by Hans Christian Graham's happy accident in a Berlin morgue.
It's not just about purple and pink on a glass slide. It's about reading the physical armor of a microscopic enemy.
It is a remarkable legacy. A simple dye showing us the very architecture of survival at the microscopic level, allowing us to choose exactly the right weapon.
And it leaves us with something pretty intense to ponder. We talked about how the outer membrane and those porin channels of gram-negative bacteria act like a fortress, blocking many modern antibiotics.
Right, the gatekeepers.
Well, as the global crisis of antibiotic resistance worsens, these bugs are rapidly evolving to mutate or shut down those porins completely to keep our drugs out. So will Graham's 140-year-old dye test soon shift from being a helpful tool that tells doctors which drug to use to a dark crystal ball that simply tells us an infection is entirely untreatable?
That is a terrifying thought.
We started in the ICU with a fighting chance, thanks to a few drops of dye. Let's hope that dye doesn't one day show us that we've run out of weapons entirely.